Namaste everyone. Today I'm before you to present list of biochemistry and biophysics important questions. In first main, some important questions are peptide chain or peptide bond. Okay. Second question, holo enzyme. Third question, active site of an enzyme. Okay. Fourth question, zwitterion. Fifth question, what are simple lipids? Give example. Sixth question, what is denaturation of proteins? Seventh question, isoelectric point. Eighth question, what is coenzyme and cofactor? Ninth question, how disulfide bond formed? Tenth question, mention any two differences between essential and non-essential amino acids. 12th question, what are polar lipids? 13th question, define hydrogenation. 14th question, rancidity. Okay, 15th question, Sanger's reagent. Okay, you need to answer all these questions for 2 marks. Now let us go through expected second main questions. Okay, you'll get the headings under second main like this. Answer any two of the following. Okay, each question carries 5 marks. In that, let us go to the first one. Explain hydrogen bond and disulfide bond. Okay. Question number 2. Explain principles and applications of HPLC. Third question. Give the reaction of amino acids with ninhydrin and Sanger's reagent. Okay. This question usually you'll get for 10 marks. Okay. And here I've asked this question for 5 marks. But I request you to consider this question for 10 marks. Okay. Fourth question, with the help of neat labeled diagram, write a short note on thin layer chromatography, in short TLC. Fifth question, explain paper chromatography. Sixth question, list the differences between fibrous and globular proteins. Or you may get the question like briefly explain fibrous and globular proteins. Okay. Seventh question, give the principle and applications of NMR. Eighth question, discuss the scope and development of biophysics. Ninth question, how are enzymes classified? Give one example for each class. 10th question, briefly give the applications of column chromatography. 11th question, what is meant by spectroscopy? Write short notes on fluorescence spectroscopy. Dear students, you will get this question even for 10 marks. Okay, keep in your mind that while answering this question, 11th question, try to answer for 10 marks too. 12th question, write the principle and applications of density gradient centrifugation. 13th question, explain the laws of thermodynamics and their limitations. 14th question, give the functions of proteins. Okay, all these questions you need to answer under second main for 5 marks. All right. Now let us go through third main. Okay, in third main, usually you'll get like answer any two of the following. Here you need to answer all these questions for 10 marks. Okay. First one, explain the factors that influence the rate of enzyme activity. Second one, explain the general characteristics of enzyme and add a note on enzyme inhibition. Third question, give the functions of proteins. The same question you may get for even 5 marks or 10 marks. By going through the marks, you must answer. Okay. Fourth question, explain primary structure of proteins. Fifth question, what is meant by spectroscopy? Write a short note on fluorescence spectroscopy. Sixth question, explain secondary structure of proteins. Seventh one, what is Michaelis-Menten equation? Mention the significance of Vmax and Km. This question you may get for even 5 marks. Okay. Eighth question, explain the properties of lipids and add a note on rancidity. Ninth question, write the following structure. First one, TAG, that is triacylglycerol, DAG, diacylglycerol, MAG, monoacylglycerol. Okay. Further, you need to write the structure of quaternary structure and you need to label. Next, the structure of alpha amino acid. You need to write the alpha amino acid and you need to label. Okay. Each structure carries 2 marks. It's very simple. You can write all this. Okay. 10th question, explain denaturation of proteins. Even I've asked this question in 5 marks. Even you'll get this for 10 marks. 11th question, write short note on GLC, that is gas liquid chromatography. Then B, scope of biophysics. Okay. Then you may get the question for tertiary structure, 10 marks. Okay. Write short notes on paper chromatography and thin layer chromatography. Each chromatography carries 5 marks each. 14th question. Write a note on principles. Okay, write a note on principle and applications of UV visible spectroscopy. Okay. 15th question. Write a short note on ultracentrifugation, then peptide bond. Okay. So this is all about the 10 marks question. Okay. Hope you have gone through all these 15 questions. Get ready for all 15 questions under third main to answer for 10 marks. Okay. Hope you don't have any kind of confusion here. Next, fourth main, answer the following in one word or sentence. Okay, each question carries 1 mark each. Okay, first question, what is saponification? Second question, what is the wavelength of light used in visible spectroscopy? Third question, expand RPM. Fourth question, what are coenzyme? Fifth question, what are simple lipids? Sixth question, define thermodynamic equilibrium. Seventh question, what is an active site? Eighth question, define isoelectric point. Ninth question, what is the unit of sedimentation coefficient? The last question, the fourth main, what are conjugated proteins? Okay. So we need to answer all these 10 questions under fourth main in one word or sentence. Okay. All the best, dear students. Hope you will be able to do well in your internal test. Thank you for watching this video.